Welcome to yet another session in zoology. In previous video, we have discussed about the chromosomal mutations based on the structure. Now we are going to continue the session with chromosomal mutations with respect to the sets of the chromosomes in the cells regarding the aneuploidy and polyploidy. Let me introduce myself. This is P. Kumari, Assistant Professor of Zoology, Tarabarma Degree College. In the previous video, we have discussed about the chromosomal mutations. Let me just brush up a little. The chromosomal mutations would be change in the chromosome structure or number. Basically, we have discussed about the changes in the structure. Changes in the chromosome structure may be due to, there were 4 different types. If a fragment of chromosome is lost, that type of mutation is referred to as deletion. If a fragment of DNA is doubled, then we call it as duplication. And if a fragment of the chromosome breaks and reattaches in a different position, in a different orientation, then that is referred to as inversion. Then a breaking of 2 segments of chromosomes from 2 different non-homologous chromosomes and then joining 2 other different non-homologous chromosomes, we call it as translocation. So this was basically changes of chromosome structure. Now today in this session, we'll be talking about chromosomal mutations based on the number of the sets. So let us get acquainted with certain terminology that we are going to use today. Haploid set as it refers to the number of chromosomes that are present within the cell with respect to the single copy of chromosomes. Then complete set or diploid set. Complete set or diploid set would mean that all the organisms have got 2 sets of homologous chromosomes and we call them as diploid. Now each type of chromosome is represented twice with an exception of sex chromosomes. Therefore, all animals and higher plants are diploid. Then coming to doubled or tetraploidy, this is where the number of chromosomes which are otherwise diploid are exactly doubled, and the total number of chromosomes what we find here is 4 times the haploid set. Then if there are more number of sets present, you call it as more number of sets. If it is more than tetraploidy, diploidy, triploidy, then you call it as polyploidy. If at all there is a loss of a chromosome or a gain of a chromosome from the diploid set, that means if it is 2n minus 1 or 2n plus 1, that is called as aneuploidy. Changes in the chromosome number occurs at the time of gamete formation during meiosis due to a very important process called as non-disjunction. So this is haploid, diploid, triploid and tetraploid slide for you. Haploid, in case here there are 3 different chromosomes, as you can see, in 3 different colors with a single chromosome representing each. When it comes to diploid, you get to have 6 chromosomes, that is each chromosome exists in pairs, a pair of pink, a pair of blue chromosomes and a pair of yellow chromosomes. And when it comes to triploid or 3n, each chromosome has got 3 copies, as you can see here. So it is 1, 2, 3 pink chromosomes, 3 blue chromosomes and 3 yellow chromosomes for our better understanding. The same thing applies to tetraploid, where the number is exactly double with the diploid. Diploid, each chromosome has got 2. In case of tetraploid, we can see that each chromosome exists in a number of 4. So here it is 4 pink chromosomes, 4 blue chromosomes and 4 yellow chromosomes. Continuing further, let us get to know about the terminology here. A bit of terminology would be always helpful. So, how do you define what is a diploid organism? So, organisms with two sets of homologous chromosomes are referred to as diploid organisms. Of course, there is an exception of sex chromosomes. Each type of chromosome is represented twice. Sex chromosomes now they can be different. As in case of humans, it is XX in females and it is XY in males. And all higher plants and animals are diploid. Coming to another terminology that is called as monoploids. Monoploids are basically are individual members of a diploid species that has only one chromosome set. That is, a monoploid is it can be distinguished it from an individual member of a normally haploid species. In order to distinguish haploid species from the monoploid members of a diploid species, we use this term as monoploid. As an example, lower plants like algae, fungi, and mosses are haploids. They have got only a single set of chromosomes in their life cycles. But male honeybees, wasps, and ants are monoploid. They develop from the unfertilized eggs by parthenogenesis. What we need to understand here, the difference between algae and the honeybees is, honeybees in the society of honeybees, the female honeybees and the worker honeybees are diploid. But when it comes to the male honeybees, they are haploid because they develop from the unfertilized eggs by a process called as parthenogenesis. Therefore, male honeybees, wasps, and ants and certain insects are referred to as monoploids. What exactly are polyploids? Polyploids are again individuals that have more than two sets of chromosomes, as in more than two sets of chromosomes, as in if it is two, you call it as diploid. Anything more than diploid would be like if it is three sets, you call it as triploids. Four sets, you call it as tetraploids. Five sets, you call it as pentaploids, so on and so forth, etc. Euploidy is another term we often come across, we use it a lot. Euploidy is referred to as, it is basically applicable to the cells and tissues of an organisms that have either the basic chromosome number of the genus or complete multiples of that particular genus, as in diploid, triploid, and tetraploid. Now, one classic example of euploidy that can be seen in humans include vertebrae and liver cell nuclei. Liver cell nuclei usually consist of 4n, 8n, or 16n chromosomal sets. And it comes to plants. In plants, we come across euploidy in stem and parenchymal tissue of apical region of flowering plants. Now, coming to the major part of the topic today, that is aneuploids. Aneuploids is supposed to be the second major category of chromosomal mutations. This involves the number and not the structure. As in previous case, we have seen about the structure. Now, an aneuploid is an individual with abnormal chromosome number that differs from the normal diploid set by one or two chromosomes. Remember, aneuploids is always a plus or minus of the diploid set of chromosomes. Here, let us talk about a few nomenclatures again. Monosomic, that means 2n minus 1, diploid set of chromosomes minus 1, that is one copy is present instead of usually two found in diploid progenitor. So, if it is in case of humans, suppose it's humans, then the 2n number of chromosomes in humans would be 46 chromosomes, and 46 minus 1 would be 45. Therefore, monosomic for humans would be 45 chromosomes, 2n minus 1. Then we have trisomic. Trisomic is where the number is 2n plus 1. So if it is 46 chromosomes, then there's one more chromosome. You add to the 10, that becomes 47 chromosomes. There is something else called as nullisomic. Nullisomic means a diploid set of chromosomes minus a pair of homologous chromosomes. So this means it's a loss of both homologous chromosomes, and generally this doesn't happen because nullisomy is lethal and such zygotes are not viable and such individuals do not survive. Now, all the sex chromosomes are aneuploids. Now, we need to understand a notation that is special notation is used to describe the sex chromosome aneuploids because it must deal with two different chromosomes. So the notations merely list the copies of each sex chromosome, such as XXY, XYY, triple X, XO, where O stands for absence of a chromosome, therefore it is just simply X. Now, this could arise, all these aneuploids result from non-disjunction of chromosomes during meiosis. What exactly is non-disjunction? Non-disjunction means not coming apart. They don't get separated, or it's a failure of chromosome pairs to separate properly during formation of gametes. Now, in the metaphase stage, we can see there are three sets of chromosomes here, and one represented in blue, the other in green, and the other in red, just for our convenience sake. So what happens is during non-disjunction, the sister chromatids they split across the kinetochore, and that is how here you find they are properly separated from each other. But when it comes to the red pair of chromosomes, you don't see them separating from each other. They don't come apart, they stay together. So when the cell divides in the center, one cell will be getting 1, 2, and 3 chromosomes. The other cell will be getting only 1 and 2 chromosomes. So this is unequal distribution of the chromosomes that happens during the non-disjunction. This could arise from a failure of homologous chromosome to separate either in meiosis 1 or the failure of sister chromatids to separate during meiosis 2 or mitosis. So once again, non-disjunction is the failure of homologous chromosomes or sister chromatids to separate properly during cell division. So here we have a pair of homologous chromosomes represented in red and blue. So when a proper non-disjunction takes place, when there is a non-disjunction, usually because this is 2, the cells are supposed to get 2 chromosomes, but here when you see there is a 2n plus 1 and 2n minus 1 because the red chromosome has separated properly, but the blue chromatids they remained together and they failed to separate. Therefore, one cell is deprived of a blue chromosome, and here it has got an extra blue chromosome. So this is about non-disjunction. So this is just for to understand exactly what we have seen so far. So we have a name, designation, constitution, and number of chromosomes. We are classifying this into euploids and aneuploids. Monoploids here, monoploids means a single set that can be represented with N, and ABC here stand for the types of chromosomes. Excuse me. And the number of chromosomes here are represented as 3. Coming to diploids, diploids have got 2 sets of chromosomes, a pair of homologous chromosomes that is represented as 2n, and therefore here each chromosome has represented in pairs 2A, 2Bs and 2Cs. So total number of chromosomes here is 6, and exactly half of the 6 would be 3, and that is what is monoploids. Coming to triploids, triploids have three chromosomes actually, one extra set of chromosomes, and therefore here you can see there are 3As, 3Bs and 3Cs, and total number of chromosomes are 9. Tetraploids will be 4N, 4 of each N, therefore 4 capital A's, 4 capital B's and 4 capital C's. This is exactly the double of diploids. That means it has got two sets of diploid chromosomes here. So the total number of chromosomes would be 12. When it coming to aneuploids, where you find a missing loss or gain of chromosomes here, the terminology we discussed was monosomic. Monosomic would be 2n minus 1, where one chromosome is lost due to non-disjunction. So here we have A, capital B, and capital C, capital C. So what happened here was the A chromosome, what was supposed to be here, is did not separate, and it has gone to the other cell, and that other cell would be exactly what you're seeing here is the trisomic. Whatever you can see here is the trisomic. So in the trisomic, it is 2n plus 1, and this is 2n minus 1. Here, one pair of homologous chromosomes is supposed to be associated with the monosomic, and so the total number of chromosomes is 5 and not 6. Now, when it comes to trisomic, in the trisomic there is an addition or a gain of chromosome, therefore one extra chromosome can be found here. So the diploid set has to be 6, so it is 6 plus 1, that comes up to be 7. Let us talk about the genetic disorders due to non-disjunction of allosomes. We all know allosomes are the sex chromosomes associated with the sex determination. The allosomes include the X and Y chromosomes in case of humans. So the first and foremost disorder due to non-disjunction that is seen in humans is called as Turner syndrome. The karyotype of the Turner syndrome is 45. So normal humans usually have 46 chromosomes, but in a Turner syndrome individual, the number of chromosomes are just 45 because there is one chromosome lost here. And these Turner syndrome individuals are generally females, and the incidence rate of the birth is 1 in 5,000 female births. So phenotype of Turner syndrome individuals are sterile females, are sterile in the sense they are not fertile and they cannot reproduce offspring. Now coming to their physical features, abnormalities developed due to a loss of chromosome is they have got a short, they are very short in stature, they have a web skin extending between the neck and the shoulders, intelligence is near normal, certain cognitive functions are defective. So this is the karyotype from a female with Turner syndrome with 45 and just one single allosome, that is X chromosome. There is supposed to be another chromosome here, either Y or X, which is missing due to non-distinction. And these are represented as the chromosomes, as you can all see, first pair, second pair, third pair, fourth, fifth, so on and so forth. And this is the 23rd pair, allosome pair, where the sex chromosomes are there, and in Turner females we can find only one X chromosome. So these are Turner syndrome females, where you can see they are short stature, then they have a typical characteristic facial feel, web skin, fold of skin around the necks. They have got heart problems, poor breast development, rudimentary ovaries, as because they are sterile, So they have underdeveloped gonadal structures. They do not undergo menstruation. Their body development is not proper. You find elbow deformity, small fingernails, shortened fingers, then widely spaced nipples across the chest, then shield-like thorax, and low hairline. All these are the abnormalities of the Turner syndrome, which are resulted due to non-disjunction of the sex chromosomes and the loss of one X chromosome. Coming to the next disorder due to non-disjunction of chromosomes is Klinefelter syndrome, as the name suggests. Karyotype of Klinefelter syndrome is 47. Here, the normal karyotype of a healthy human individuals is 46, but we find it as 47 because this is 2n plus 1. So they have got 44 chromosomes, that is 22 pairs of the autosomes are normal, but when it comes to the sex chromosomes, there is an extra chromosome here, as we can see, and that is XXY. Incident rate is 1 in 1000 births, and this is the trisomy of the 23rd pair, where the 23rd pair fails to segregate during cell division. The phenotypes of Klinefelter syndromes are male, and you can find a barred body. Barred body is nothing but it is a condensed X chromosome, highly condensed and darkly stained X chromosome in the nucleus. Now that the males, Klinefelter males have got two copies of X chromosomes, you expect a barred body. Now they appear physically normal until they attain puberty, and primary features include lanky body, sterility they are not fertile, less body hair, there is breast growth. Probably this is because of the two X chromosomes that are present, and this feature is called as gynecomastia. Then small tentacles we can notice. Testicular size is very small. Then intelligence is usually normal, but they have got problems are reading and learning difficulties with problems with speech is normally observed in Klinefelter syndrome males. So let's look at the karyotype of the Klinefelter syndrome means all the other sets of the autosomes appears to be normal, but when it comes to the 23rd pair, we find an extra copy of X chromosome which failed to separate during the cell division due to non-disjunction, and therefore we have two X chromosomes along with the Y chromosome. And this is probably Klinefelter syndrome male looks like. There's a lack of beard or body hair, then enlargement of breast that is called as gynecomastia, long arms, long bodies. This is in contrast to the Turner syndrome where the body stature is very short. Then testicular atrophy, no proper development of the gonads, leading to the infertility, lack of pubic hair and hip-like female structures. So basically we are finding more of female qualities here because of the presence of an extra X chromosome. Next, we have got the other abnormal conditions include XYY males and triple X females. Generally, these two people are fertile. So, XYY males are fertile and triple X females are also fertile. Meiosis shows normal pairing of X with one of the two Ys, and the other Y does not pair, and it is not transmitted to the gametes in case of XYY. Therefore, the gametes contain either X or Y chromosomes, and there is never possibility of a YY or XY chromosomes in the gametes. So therefore, XYY males are always fertile, and this seems to be normal. The X trisomics, this is X trisomics, are phenotypically normal, and these are fertile females. Meiosis shows pairing of only two X chromosomes. The third one does not pair, hence the eggs bear only one X, and like that of XYY males, the condition is not passed on to the progeny. And therefore, the triple X females or XYY males, they appear to be normal in spite of non-disjunction. So this is about the genetic abnormalities of uh, allosomes. And let us talk about the genetic disorders due to non-disjunction of autosomes. Autosomes are supposed to be the remaining 22 pairs of the chromosomes in the humans. So the first and the foremost disorder associated with the non-disjunction of autosomes is the Down syndrome. Karyotype is 47, excuse me, 47. That means it has got 45 uh, chromosomes plus an extra pair of X chromosomes, that is XX or XY. And this is because of trisomy of 21st pair of autosomes. Now, phenotype, it includes mental retardation. Person is generally mentally retarded. They have a broad, flat face, eyes with an epicanthic fold. They are short in stature. They have short hands with a crease across the middle, a large wrinkled tongue. All these are the physical characters, phenotypic expressions of a Down syndrome individual. Females may be fertile and may produce normal or trisomic progeny. Mm, but males are sterile with very few exceptions. Mean life expectancy is about 17 years, and only 8% of the persons with the Down syndrome survive the past age of 40 years. Therefore, the lifespan is very short. The only other human autosomal trisomics to survive to birth are those with the trisomy of 13 chromosome and trisomy of 18 chromosome. Both have severe physical and mental abnormalities. All other trisomics, they die in the utero or within the uterus even before the birth of the infant. So one such abnormality, what we can see is yeah, this is a Down syndrome karyotype, as we can see all the chromosomes here. This is the 21st pair of autosomes where you find three chromosomes here. Normally there has to be a pair, but here it failed to separate during the non-disjunction and during the cell division, and because of the non-disjunction and extra chromosome happens to remain here, and because of this trisomy of 21st chromosome, Down syndrome is possible. Then we have yet another trisomy of 18th chromosome, that is Edwards syndrome. The karyotype is 46 chromosomes XX or 46 chromosome XY. So Edwards syndrome can be a male or a female, as in this case, unlike the previous cases. And the extra chromosome would be trisomy is with the 18th chromosome. It is a slow intrauterine growth and a low birth weight. These individuals show very slow development within the uterus, and they have a very low birth weight. Affected individuals may have heart defects and abnormalities of other organs that develop before birth. Other features also include small abnormal head, abnormally small shaped head, and they have a small jaw and mouth, and clenched fists. Their hands are always clenched with the overlapping fingers, and due to the presence of these severe life threatening medical problems. Many individuals with trisomy of 18, they die either before birth or within their first month of birth. So this is the karyotype of Edwards syndrome. This is in case of a male because it is XY. Now, the trisomy is due to the 18th of autosome, where you find 3 copies of 18th chromosome here. So this is Edwards syndrome. The rest of the chromosomes, they exist in homologous pairs, just two, including the sex chromosomes are normal here, X and Y. If it is X and Y, it would be an Edwards syndrome male. If it is XX, it would be an Edwards syndrome female. So this is a trisomy of 18th chromosome. Then we have Patau syndrome. Next genetic disorder associated with the autosomes. Karyotype is 46 chromosomes in males or 46 chromosomes in females. Uh, and this is trisomy of the 13th chromosome. Patau syndrome is associated with the severe intellectual disabilities and physical abnormalities in many parts of the body. Individuals with trisomy 13 often have heart defects, brain or spinal cord abnormalities. Then they have a very small and poorly developed eyes, what we call it as ophthalmia, that is a very small and poorly developed eyes. Then they have a very weak muscle tone called as hypotonia, and then they have extra fingers, extra toes, and there is an opening in the lip called as cleft lip, with or without an opening in the roof of the mouth, that is called as cleft palate. Then this is what are the problems these Patau syndrome individuals face. Now, due to the presence of these several life-threatening medical problems, many infants of trisomy 13 die within their first days or first weeks of life. So this is a Patau syndrome karyotype, as you can see. All the other rest of the autosomes have got just two homologous chromosomes. When we look at the 13th chromosome, we find an extra chromosome here which failed to separate during a cell division. So Patau syndrome can be males or females with this problem. And this is a Patau syndrome male child. You can see the clenched hands here. It's always clenched, the fists are held tightly, and you can find polydactyly or extra fingers. Then the undescended or abnormal testes. When you look at the ears are malformed ears, then it's a small head, eyebrows are absent, there is a cleft lip or a cleft palate inside the mouth. Usually these individuals, they hardly survive very first few days of their life. Now let's talk about polyploidy. Polyploidy is very common in plants but rarer in animals. An increase in the number of chromosome sets has been an important factor in the origin of new plant species. So polyploidy is basically of two types, autoploidy and autopolyploidy and allopolyploidy. Autopolyploidy is where all chromosome sets are from the same species, where autoploidy is where all the chromosome sets are from the same species, uh, and they contain multiple sets which are identical to the parent species. Um, Alloploidy, in other words, means different. Allo means different. So chromosome sets are from two or more species containing two or more copies of each of the input genomes. So hybrids, basically autopolyploids are hybrids of two or more species. Now, triploids or autopolyploidies can arise in a number of ways. You can produce them in a number of ways. So the failure of all chromosomes to separate uh, or segregate during meiosis gives rise to a diploid gamete, which, when fertilized by a haploid gamete, uh, results in the zygotes that have three sets of chromosomes. So we have a diploid gamete which is fertilized with an haploid gamete, results in autotriploids. Diploid gamete again is resulted due to non-disjunction, where diploid set of chromosomes will become haploid in the gametes. So 2n plus n would result in 3n. Or else a triploid zygote may result due to a chance fertilization of an ovum by two sperms. Either way, this also can lead to the formation of autotriploids. Or else, triploids can be generated experimentally by crossing diploids with tetraploids. This results in triploids. Um, they arise spontaneously in nature, um, but they can be constructed by geneticists from the cross of a tetraploid and a diploid. The 2n and the n gametes produced by the tetraploid and the diploid respectively unite to form a triploid. Triploids are characteristically sterile. So the bananas and the watermelons that are used widely available commercially are sterile triploids without seeds. Economically important triploid plants are propagated asexually and include Solanum, tuberosum, etc. So the flower and fruit of polyploidy plants are larger in size than the diploids, and tetraploids and octoploids are commercially available. So these are the watermelons without seeds that are commercially available, and these are uh, triploid watermelons, and these are sterile, and so you cannot expect the next generation of plants from this watermelon seeds. Now these are strawberries, what you see here. This smaller one is a diploid strawberry, but here what you're seeing is octoploid strawberry. So these strawberries is an octoploid. Generally, the autopolyploids are larger in size than the diploids. We all know that. The flower and the fruit of polyploid plants are larger in size, increasing their horticultural commercial value. So this strawberry is octoploid strawberry, and this is a diploid strawberry. Now, what we are finding here are the tetraploid grapes and the diploid grapes. So these are the diploid grapes with a normal size, and next to them we can see the tetraploid grapes which are larger in size. So how do we get these autotetraploids, or can they be synthesized experimentally? Yes, this can be done. What you're seeing here is a normal branch with a diploid number of chromosomes here, and this is the terminal part. So what actually happens here is they autotetraploids arise by the doubling of 2n complement to the 4n diploid to the tetraploid. This doubling can occur spontaneously, but it can also be induced artificially by applying chemical agents that disrupt microtubule polymerization. A commonly used anti-tubulin agent is colchicine. In colchicine-treated cells, the chromosomes are doubled in S phase of the cell cycle, but chromosomes are not segregated, and a nuclear membrane is formed around the entire doubled set of chromosomes. Thus, treating diploid cells with colchicine for one cell cycle leads to tetraploids. with exactly 4 copies of each type of chromosome. Treatment for another additional cell cycle produces octoploids and so far and so forth. So for this particular terminal branch, we are applying colchicine is applied to the terminal bud of this branch. Now the cells are tetraploid. So what happens here is the chromosomes, they double, they do not segregate, and the nuclear membrane is formed around the entire doubled set of the chromosomes, and that leads to the formation of the tetraploids. So the cells are tetraploid and the leaves and the fruits are larger for this branch. Next thing what you do is cutting off the tetraploid branch will produce tetraploid plants with tetraploid foreign of flowers, fruits and seeds. This is how it can be artificially done, synthesized, and this is how you can produce autotetraploids from a diploid plant. So this is what is happening. Autopolyploidy results in offspring with 2 sets of chromosomes. So this is what is happening. This is a diploid parent. You induce or treat with this chemical called as colchicine, and during the cell cycle what happens is this diploid will undergo duplication, replication, and it produces a set of 4 tetraploid chromosomes. Now these usually have to segregate during the cell division, but that doesn't happen. Instead, the nuclear envelope develops, and therefore this particular part or this particular cell or tissue would have all the 4 sets of the chromosomes of the parent. So this is how autopolyploidy is done. Next topic would be allopolyploidy. An allopolyploid is a plant that is a hybrid of 2 or more species containing 2 or more copies of each of the input genomes. Allopolyploidy has been important in the production of modern crop plants. New World cotton is a natural alloploid that arose spontaneously, as exactly is the wheat. So, allopolyploids are also synthesized artificially to combine the useful features of the parental species in one type. Only one synthetic amphidiploid, that is double diploid, has ever been widely used commercially, is a crop known as triticale. This is the crop known as triticale, and this triticale is an amphidiploid between the wheat, which has got 42 chromosomes, and the ray plant that has got 14 chromosomes. Hence, for a triticale, it is amphidiploid, double diploid. So it is 2n into 21 plus 7, that makes it 56, actually, not 52, it has to be 56. So this novel plant combines the high yields of wheat with the ruggedness of ray. So this is the triticale plant that I would like to show to you. So this is where the wheat production is done. Then polyploidy in animals. Polyploidy is more common in plants than in animals, but there are some naturally occurring polyploid animals. Polyploid species of flatworms, leeches, brine shrimps, they reproduce by parthenogenesis. Polyploid species uh, like triploid and tetraploid Drosophila have been synthesized experimentally. They are occurring naturally occurring polyploid amphibians like frogs and toads that can reproduce sexually. Then we have polyploidy salamanders and lizards that reproduce parthenogenetically. We have what is called as triploid oysters and diploid oysters. The sterility of triploids have become commercially exploited in animals as well as in plants. Triploid oysters have been developed because they have a commercial advantage as they are palatable in the sense they are edible over their diploid relatives. The diploids, they go through a spawning season when they are unpalatable, but the sterile triploids do not spawn and are palatable year round. So this is how triploid oysters have more commercial advantage than their diploid uh, counterparts. So here on this we have a diploid and a triploid oysters. So basically there's a structural difference. They are larger, and these diploid oysters are edible and they're available throughout the year because they do not undergo spawning. They do not reproduce as they are sterile. So with this we conclude about the chromosomal mutations based on the structure and number. Hope this will be useful to you people. Thank you. Ni engkama?